Hi there, friends! Today, we'll do a fun science story about how tiny bacteria can help plant change and grow better. Our activity is called genetic transformation. Sounds big, but don't worry, it just means adding something new to a plant to make it stronger and healthier.
Meet our little helper, agrobacterium. It's a very tiny living thing that lives in the soil. If a plant gets a small wound, this bacterium comes close and puts a piece of its own DNA inside the plants. The DNA is like a tiny message telling the plants what to do. For example, make more vitamins or be strong against bugs. That's why agrobacterium is sometimes called nature's little engineer. In this activity, we don't use real bacteria. Instead, we'll use crafts and models to pretend. Here's what we need. One big plastic box for the plant cell, one small box for the bacterial cell, color paper, scissors, tape, and glue, some beads or buttons, and a small envelope called the transformation toolkit. Red paper for the special DNA piece that goes into the plant. Blue paper for helper parts. Green for food making. And yellow for copying the circle. Glue or tape them into a big ring. That's your DNA circle. Now, talk with your group. Which part goes into the plant? What happens if one part breaks? And how could we change this DNA circle to help the plant grow better?
Phase 2. The transformation game. Put 10 beads in the plant box. These are the plant's chromosomes or tiny parts that hold its instruction. Place your DNA circle inside the smaller box. That's the bacteria. Then keep them about 1 ruler apart.
Time to start the signal round. Each group takes turns drawing plant signal cards. The cards might say things like temperature change, sugar, and nutrient signal. When the right signal appears, move your bacteria box closer to the plant box, just like real bacteria moving towards a plant's wound. Next, we'll do the transformation steps. Step 1. The bacteria attaches to the plant. Step 2. It makes special helper parts. Use beads to show this. Step 3. Cut out your red paper piece. That's the DNA being sent. Step 4. Using a paper roll to move it into the plant box. Now, your plant has new DNA inside. Finally, connect it with the plant's beads. This shows the new DNA joining with the plant's own. Now, let's stop and think. Ask your group, which DNA part was moved? Where did it join the plant's DNA? What new thing might happen to the plant? Maybe it can make more vitamins, fight off bugs, or grow faster. Finally, let's compare. There are other ways to move DNA, like the gene gun, which shoots it in, or using electricity to open tiny holes in the cell. But agrobacterium is gentle and natural. It's been doing this all by itself for millions of years.
And that's the end of our lab. Let's remember what we learned today. Tiny bacteria can help plant and grow stronger. We can model this safely with paper and beads. This is how science helps us make crops that can feed more people and stay healthy. Science is amazing, right? Grabe.